Hello and welcome back to NEET Prep Go. So today we'll be doing another chapter, Microbes in Human Welfare. So without any further ado, let's get started. Besides microscopic plants and animals, microbes are the major component on this earth. Major component of biological systems on this earth. So, besides for macroscopic, like big plants and animals, microbes are also major component of our biological system on this earth. Microbes are present in soil, water, inside our bodies, and that of the bodies of other plants and animals. They are present even at sites where no other life form can exist. You saw archaebacteria, right, in kingdom Monera. That is, they can exist in sites such as deep inside the geyser, wherein the temperature can be as high as 100 degrees Celsius in thermal vents, etc. Deep in the soil, in many layers of snow, and in highly acidic environments also. And microbes are diverse, that is protozoa, fungi, bacteria, microscopic plant and animal viruses, viroids, and prions, which are proteinaceous infectious agents. That is prions, they are folded proteins. They cause mad cow disease in cattle, and its variant in humans is CR Jacobs disease. Now, microbes like bacteria and many fungi can be grown on nutritive media to form colony that can be seen with the naked eye. Such cultures are useful in studies of microorganisms. So see, first is rod-shaped bacteria, Bacillus plural bacilli in 1500x under a microscope. And second is the spherical bacteria, that is coccus or plural cocci, also in 1500x. Then the third one is a rod-shaped bacterium, that is Bacillus, having flagella, but this is in 50,000x. So each single one is over here with flagella. It is rod-shaped bacterium Bacillus in 50,000x. This is how it looks. Then over here we have firstly a bacteriophage. What is a bacteriophage? A virus that attacks bacteria. This is a bacteriophage. Bacteriophage is a virus which attacks bacteria. What all does it have? Firstly, it has a head, then collar, then tail, then plate, then the plate has pins, and then lastly prongs. Head, collar, tail, plate, pins, prongs. Okay. Then second is a picture of adenovirus, and what does adenovirus cause? It causes respiratory infection. So this is a picture of adenovirus. And lastly, this is a picture of 1 lakh to 1,50,000 x magnification of TMV, tobacco mosaic virus. Look how it looks. Compact rod-shaped virus, tobacco mosaic virus. Now, these are two colonies. First is a bacterial colony. This is how it looks, and the second is a fungal colony. Both are grown in a petri dish. So we know that we read in Human Health and Disease that microbes cause diseases in human beings, but they all and also in animals and plants. But this doesn't mean that all the microbes are harmful. Many of them are important to us in many ways. Let's discuss them below. So firstly, microbes in household products. So what is the most common microbe which is used in our household product? So that is the most common example is the conversion of milk to curd. That is, how do we convert milk to curd? With the help of the bacteria lactobacillus and other bacteria which convert milk to curd. That is, the entire group is called as lactic acid bacteria or lab. They grow in milk and convert it into curd. How do they convert it into curd? They produce acids which coagulate and partially digest the milk proteins. That is, the lactic acid bacteria or lab, out of one of which is lactobacillus, namely, they convert milk to curd. How do they convert milk to curd? They produce acids. These acids coagulate and partially digest the milk proteins. So if a small amount of curd is added to the fresh milk as inoculum or starter, so if we take a small amount of curd, so do you every day go and buy fresh curd? No, a lot of people do not do that. A lot of people, they buy milk regularly, and they once in a while, they let's say twice in a week maybe, they will buy curd, a small cup of curd. Then they will take 1 spoon of or 2 spoons of curd, they will put it in the milk, and then overnight they will the entire milk will get converted into curd. So therefore therefore a small amount of curd is added to fresh milk as inoculum or starter, as a starter basically, or inoculum you call it, and it contains millions of lactic acid bacteria. That small amount only causes contains millions of lactic acid bacteria, which at suitable temperature they multiply, and therefore they convert milk to curd, which also improves its nutritional quality, because curd has, that is from milk increases vitamin B12, and in our stomach too it plays very beneficial role in checking disease causing microbes. So it improves vitamin B12, and in our stomach also it plays very beneficial role in checking disease causing microbes. Then in dough, that is which is used to make food, as well as dosa, idli and everything, that is there is a puffed up experience is also fermented by bacteria. The puffed up appearance of dough is due to the production of carbon dioxide gas. So firstly, the dough which is used for making foods, it can be fermented by bacteria also, but at the same time it is usually fermented by yeast, baker's yeast. What is the baker's yeast called as? It is called as Saccharomyces cerevisiae. Saccharomyces cerevisiae. Toddy, which is a very traditional drink in South India, in some parts of South India, is made by fermenting sap from palms. This is also very important. Then microbes are also used to ferment fish, soya bean, bamboo shoots to make food. Cheese is one of the oldest items in which microbes were used. Different varieties of cheese are known by the characteristic texture, flavor, taste and specificity coming from the microbes which are used. So for example, the Swiss cheese has large holes which are formed due to carbon dioxide, and who forms these carbon that is who leads to such a huge amount of production of carbon dioxide to form such big holes in Swiss cheese? It is due to a bacterium called as Propionic bacterium Sharmani. Because of this, that is production of large amount of carbon dioxide takes place by a bacterium known as Propionic bacterium Sharmani, because of which these large holes are formed in Swiss cheese, and that's why. So to make Swiss cheese, Propionibacterium sharmani is used, and Roquefort cheese is made or ripened by growing a fungi, specific fungi on them, and this gives them a particular flavor. Now microbes in industrial products. Even in industry, microbes are used to synthesize a number of products valuable to human beings, but on an industrial scale, the beverages and antibiotics produced at an industrial scale, they can't be made like this only simply. They require very large vessels for fermentation to take place, and these large vessels are called as fermenters. These large vessels are called as fermenters. Now, fermented beverages. Microbes, especially yeast, have been used immemorial for the production of beverages. What kind of beverages? Wine, rum, whiskey, brandy, and beer. Wine, beer, whiskey, brandy, rum. For this purpose, what is used? Saccharomyces cerevisiae. The same one is used, and which was used for bread making, and it is called as brewer's yeast. So, Saccharomyces cerevisiae. It is also called as baker's yeast, because it is used for bread making, dough and all, make cakes and all, and it is also called as for the puffed up appearance, and it is also called as brewer's yeast. Why? Because it is used to make beverages such as whiskey, brandy, rum, beer and wine. Now it is used fermenting malted cereals, fruit juices to produce ethanol. So it is used to ferment malted cereals, fruit juices to produce ethanol. Depending on the type of raw material which is used for fermentation, with or without distillation, different types of alcoholic drinks are obtained. So wine and beer can be made without distillation. You don't need distillation for wine and beer. Whereas other, that is rum, brandy and whiskey, they are produced by distillation of fermented broth. Now antibiotics. Antibiotics are produced by microbes are regarded one of the most significant discoveries of 20th century, because why? Because they have greatly contributed towards the welfare of the society. Antibiotics in Greek, that is anti means against and bio means life, so therefore against life, but in the context of disease causing organism. For human beings they are pro-life, but whereas for bacteria they are anti-life, that is against life. Antibiotics are chemical substances which are produced by some microbes, and they can kill or retard the growth of other microbes. So, so antibiotics are substances which are produced by one group of microbes, and they can kill or retard the growth of other disease-causing microbes. So you are familiar with the commonly used antibiotic penicillin. Alexander Fleming was once working on Staphylococcus bacteria when he observed a mold growing on one of his unwashed culture plates, around which Staphylococcus could not grow. So by accidentally, this was penicillin was an accidental discovery. How? That is, one day Alexander Fleming who was growing, that is, he was experimenting on Staphylococcus bacteria, and he was growing them, but in one of his culture plates he had not washed it, and a mold was growing in one of those culture plates, because you know fungi grow in moisture and in dark and moist regions. So he found that Staphylococcus would not grow around the mold, and this is because the mold produced a chemical called as penicillin, which he named it after the mold Penicillium notatum. However, its full potential as an effective antibiotic was established later on by Ernst Chain and Howard Florey. The antibiotic was extensively used to treat American soldiers in World War II. So Alexander Fleming, Ernst Chain and Howard Florey, all three were given Nobel Prize in 1945 for this discovery. After penicillin, other antibiotics were also purified from microbes, and such as ampicillin, tetracycline, etc. Antibiotics have greatly improved our capacity to treat deadly diseases such as plague, whooping cough, or kali khasi, diphtheria or galghotu, and leprosy or kast roag. So because of antibiotics, we can treat a lot of deadly diseases such as plague, which is caused by rat flea, or Yersinia, that is rat flea is the vector, Yersinia is the causative organism. Then whooping cough, also called as kali khasi. Why is it called whooping cough? Because it is mostly seen in children less than 5 years of age, and when they cough they produce a whoop like sound, therefore it is called as whooping cough. It is also called as kali khasi. So due to antibiotics we can treat successfully deadly diseases which used to kill many people at a time earlier. Now we can easily treat them. They are plague, whooping cough or kali khasi, then galghotu or diphtheria, and kast roag or leprosy. Now chemicals, enzymes and other bioactive chemicals, enzymes and other bioactive molecules. Microbes are also used for commercial and industrial production of certain chemicals like organic acid, alcohol and enzymes. What are the examples? Again, this is very important. That is, acid producers, firstly Aspergillus niger, which is a fungus of citric acid. Aspergillus niger is a fungus of citric acid. Then Acetobacter aceti is again a bacterium of acetic acid. So Aspergillus niger is a fungus of citric acid, and Acetobacter aceti is a bacterium of acetic acid. Clostridium botulicum is a bacterium of butyric acid, and Lactobacillus is a bacterium of lactic acid. So, microbes are also used for commercial and industrial production of certain chemicals such as organic acid, alcohol and enzyme. So first, organic acid, that is Aspergillus niger or niger. What is it? It is a fungi, and what does it produce? It produces citric acid. Then secondly, Acetobacter aceti. What is it? It is a bacterium. What does it produce? Acetic acid. Then Clostridium butylicum. Clostridium butylicum. That is the spelling, right? Correct. Butylicum. What does it? It is. This is also a bacteria. What does it produce? It says butyric acid. And lastly, which we all know, lactobacillus. And what does it produce? Lactic acid. So 4 organic acids: Aspergillus niger, which is a fungi. Rest 3 organic acids, they are produced by bacteria. Aspergillus niger is a fungi and it produces citric acid. Then secondly, Acetobacter aceti is a bacterium which produces acetic acid. Clostridium butylicum is a bacteria again and it produces butyric acid. And Lactobacillus is another bacteria and it produces lactic acid. Yeast, Saccharomyces cerevisiae, also called as baker's yeast, also called as brewer's yeast, is used for commercial production of ethanol. It is used for commercial production of ethanol. It is used for commercial production of, that is, bread making, cake, pastry, and all that. It is also used to commercially to make beverages such as wine, beer, whiskey, brandy, and rum. So there are many uses of yeast. Now microbes are also used for production of enzymes, lipases. So microbes are also used for the production of enzymes. So till this was organic acid, then here came yeast, which already you had learned. From here, it is the production of enzymes. So lipases are used in detergent formulations, and they are helpful in removing oily stains from laundry. Then you must have noticed that bottled fruit juices brought from the market are clearer as compared to that made at home. This is because they are clarified by the use of pectinases and proteases. Then lastly, streptokinase is produced by the bacterium Streptococcus and modified by genetic engineering. It is used as a clot buster for removing clots from blood vessels of patients who have undergone myocardial infarction or heart attack. Okay. So now let's come to enzymes. So lipases, microbes are also used for the production of enzymes. So firstly, lipases. What are lipases used? They are used in detergent formulations and they help to remove oily stains. Detergent formulations, and they remove oily stains. Lipases are used in detergent formulations and they remove oily stains. Then second is pectinases and proteases clarify bottled juices. Pectinases and proteases, they clarify bottled juices. Clarify bottled fruit juices. Then streptokinase, which is produced by Streptococcus. So then thirdly, streptokinase, which is produced from bacterium Streptococcus. It is used as a clot buster. It is used as a clot buster for removing clots from the blood vessels of patients who have undergone myocardial infarction, which leads to heart attack. So basically all those people who have clots in their blood vessel, let's say someone has a clot in their aorta or something, so then streptokinase is used as a clot buster to remove this clot so that blood can flow. Otherwise, so those people will be having a heart attack when not cardiac arrest. Remember, cardiac arrest, you must have learned in circulation, that is cardiac arrest is the stopping, abruptly the heart stops, whereas in heart attack it's due to a blockage. Slowly the heart muscles will not be receiving enough blood, and when they won't receive enough blood, then that blood has oxygen, so basically they won't be receiving enough oxygen, so those tissues which do not receive oxygen, they die, and that is called as myocardial infarction, and due to this myocardial infarction it leads to heart attack. Another bioactive molecule is cyclosporin A, which is used as an immunosuppressive agent in organ transplant patients. So cyclosporin A is used as an immunosuppressive agent. So basically there is graft versus host diseases there, right, wherein after a transplant, that is the body tries to attack it because it realizes that it is not his own DNA or its own it is not its own organ, so basically it tries to that is, it recognizes the transplant as a foreign material and it rejects it, and therefore there is immune reaction in WBC and everything against the that organ. So therefore cyclosporin A is an immunosuppressive agent, and the organ transplant patients have to take it their entire life, and it is produced by a fungus, Trichoderma polysporum. So firstly we saw organic acid, citric acid, butyric acid, lactic acid, and acetic acid. Then we saw enzymes, that is lipases, pectinases, proteases, and streptokinase. Now we are seeing streptokinase is a bioactive molecule rather, and now we are seeing another bioactive molecule, that is cyclosporin A, which is produced by Trichoderma polysporum, which is used as an immunosuppressive agent in organ transplant patients, and it is produced by a fungus. Then statins produced by the yeast Monascus purpureus have been commercialized as blood cholesterol lowering agent. It acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. So statins are produced by the yeast Monascus purpureus. Statins is produced by another yeast called as Monascus purpureus, and it has been commercialized as blood cholesterol lowering agent. It acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. So it is a blood cholesterol lowering agent because it acts by competitively inhibiting the enzyme which is responsible for the synthesis of cholesterol. Now, microbes in sewage treatment. We know that large quantities of wastewater are generated every day in cities and towns. A major component of this wastewater is human excreta. The municipal wastewater is also called as sewage. The municipal wastewater is called as sewage. It contains large amounts of organic matter and microbes, and many of this are pathogenic. Have you ever wondered where this huge quantity of sewage or urban wastewater is disposed of daily? It cannot be discharged into normal water bodies like rivers and streams. You can understand why. Obviously, because it contains so many pathogens and so many things. How can you just dispose it of human excreta and all? So before disposal, it is treated in sewage treatment plants, STP, to make it less polluting. Treatment of wastewater is done by heterotrophic microbes naturally present in the sewage. This treatment is carried out in two stages. So firstly, primary treatment. Primary treatment, that is these treatment steps basically involve the physical removal of particles large and small from the sewage through filtration and sedimentation. These are removed in stages. Initially, floating debris is removed by sequential filtration. So first in sewage, that is sewage treatment plants, that is municipal wastewater is called as sewage. So in sewage treatment plant, firstly there is primary treatment. In primary treatment involves the physical removal. What is primary treatment? It involves the steps basically involve the physical removal of particles which are large and small from the sewage through filtration and sedimentation. First you filter, then sedimentation, that is, as a precipitate it will sediment down below and then you can remove it. So these are removed in stages. Initially the floating debris is removed by sequential filtration. So by sequential filtration the floating debris is removed. Then grit. that is soil and small pebbles, are removed by sedimentation. So soil and small pebbles, they will settle down below and they can be removed as sedimentations. Then sedimentation, then all the solids that settle down form the primary sludge. All the solids which settle down are form the primary sludge, and the supernatant forms the effluent. One that doesn't settle down, that is, it forms the effluent. The effluent from the primary settling tank is then taken to the secondary treatment. So, firstly, by sequential filtration, the floating debris was removed. So, there was floating debris, and this floating debris was removed by sequential filtration. Then sedimentation took place, wherein the grit, that is the soil and pebbles, they all settled down below, and these were removed by sedimentation. That is, after they were sedimented, they formed the primary, and all the solid, they formed the primary sludge, and the one that was remaining, that is the supernatant, it formed the effluent. Now this is which is called as the effluent. This effluent from the went from the primary settling tank to the for secondary treatment. That is secondary treatment, or it is also called as biological treatment. So first is primary treatment, second is secondary treatment. Secondary treatment is also called as biological treatment. The primary effluent is passed into large aeration tanks where it is constantly agitated mechanically and air is pumped into it. This allows vigorous growth of useful aerobic microbes into flocs, that is masses of bacteria associated with fungal filaments, to form mesh-like structures. While growing, these microbes consume the major part of the organic matter in the effluent. This significantly reduces the biochemical oxygen demand, or BOD, of the effluent. So first is primary treatment. In primary treatment, that is, the large and small particles are physically removed. Then second, through filtration and sedimentation, that is, initially the floating debris, by sequential filtration it is removed. Then the grit, that is the pebbles and soil, they are removed by sedimentation, that is, they settle down. And this as the pebbles and the soil, it settled down, that is called as the primary sludge. And the supernatant, that is, which is formed above the primary sludge, it forms the effluent. Now the effluent is taken to from the primary treatment, it is now transferred to secondary treatment. The secondary treatment is also called as biological treatment. The primary effluent is passed into large aeration tank, where in aeration tank, that is, constantly the air is pumped into it, and it is constantly agitated mechanically. This allows vigorous growth of useful aerobic bacteria into flocs. What are flocs? They are masses of bacteria associated with fungal filaments, which form a mesh-like structure. While growing, these microbes consume the major part of the organic matter in the effluent, and this significantly reduces the BOD, biochemical oxygen demand, of the effluent. So the effluent requires the oxygen, right, for the microbes to grow, that is the aerobic and harmful microbes basically. So the BOD, that is biochemical oxygen demand, is significantly reduced because in the large aeration tanks, where air is constantly pumped and mechanically agitated, there is vigorous growth of useful aerobic microbes into flocs, that is microbes which are joined by fungal filaments, and they consume the major part of the, that is, they consume the oxygen because they require, the aerobic microbes require the oxygen. So biochemical oxygen demand of the effluent is reduced, and it refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. What is BOD? Biochemical oxygen demand refers to the amount of oxygen which would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. So the amount of oxygen which would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria, then that is called as biochemical oxygen demand. Now the sewage water is treated till the biochemical oxygen demand is reduced. The biochemical oxygen demand test measures the rate of uptake of oxygen by microorganism in a sample of water, and thus indirectly biochemical oxygen demand is a measure of the organic matter present in the water. The greater the biochemical oxygen demand of the water, more is the polluting potential. So basically, if you put some if you put a lot of bacteria in, let's say, 1 liter of water, then the amount of oxygen that can be consumed, if all the organic water, that is, if all the organic matter in 1 liter of water were oxidized by bacteria, is called as biochemical oxygen demand. The greater the biochemical oxygen demand, the more will be the pollution, because that means there are more and more microbes which are utilizing this oxygen. So basically, due to the flocks, the BOD of the effluent becomes lesser and lesser because they consume all the oxygen. Then once the BOD of the sewage wastewater is reduced significantly, the effluent is then passed into a settling tank where the bacterial flocks are allowed to sediment. So now first you form the bacterial flocks, which that is flocks. Why? Because the bacteria or the microbes, that is, they are connected by fungal filaments. So now the flocks, bacterial flocks, they were formed due to in the aeration tank, and they consumed up all the oxygen, and the biochemical oxygen demand of the effluent now reduced significantly. Now since it has reduced, now from the aeration tank, this wastewater goes to the settling tank, where these bacterial flocks, which reduce the biochemical oxygen demand significantly, they are allowed to sediment. This sediment is called as activated sludge. So the earlier sediment, which was made up of grit, that is, pebbles plus soil, was called as primary sludge. Whereas now the sediment which is made, where the bacterial flocks are allowed to sediment, so these bacterial flock sediment is called as activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as inoculum. So now you saw what is inoculum or starter, right? Where you used to take 1 or 2 spoons of curd and used to put it in milk, and they used to multiply rapidly at the present, that is, suitable temperature. And also similarly, these small amount of this activated sludge, which contains flocks of bacteria, it is put in it is pumped back into the aeration tank, and it serves as inoculum or starter. So if you put little bit of flocks and all, since there is a lot of oxygen and in the aeration tank, and it is mechanically being pumped, then more and more flocks of bacteria will grow, and they will consume more and more oxygen, since the BOD of the wastewater will be high, and they will significantly reduce it. Then they will again come over to the settling tank, and they will settle down. Then as activated sludge, again little bit will go back, so this will keep going. Then the remaining major part of the sludge, that is activated sludge, is pumped into large tanks called anaerobic sludge digesters. Here other kinds of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge. During the digestion, bacteria produce a mixture of gases such as, so the activated sludge, some part of it is served as inoculum and pumped back into the aeration tank, but majority of it, or remaining major part of the sludge from the settling tank, is pumped into large tanks called as anaerobic sludge digesters. These anaerobic sludge digesters, here other kinds of bacteria which grow anaerobically digest this flock of bacteria and fungi which was there in the sludge. And during this digestion, these anaerobic bacteria, they produce a mixture of gases such as CH4, methane, hydrogen sulfide, H2S, and CO2, carbon dioxide. These gases form biogas, and they can be used as a source of energy as it is flammable. So in many places where near biogas plants are there, that is, in your cooking, biogas is used. The effluent from the secondary treatment plant is generally released into natural water bodies like rivers and streams. So now the effluent from secondary treatment, the effluent that we get from the secondary treatment plant, is generally released into natural water bodies like rivers and streams. An aerial view of such a plant is shown in figure 10.7. So this is an aerial view of the sewage plant. You can appreciate how microbes play a major role in treating millions of gallons of wastewater every day across the globe. This methodology has been practiced for more than a century now in almost all the parts of the world. Till date, no man-made technology has been able to rival the microbial treatment of sewage. So there has been no as such man-made technology in which, that is, we use in sewage wastewater treatment plant, a major or chunk or almost more than that is 80% of the work is done due to microbes, that is reducing the biochemical oxygen demand and all. So this has been possible due to microbes, and this methodology has been practiced for more than a century now, and no other man-made technology has been able to rival the microbial treatment of sewage. You are aware that due to increasing urbanization, sewage is being produced in much larger quantities than ever before. However, the number of sewage treatment plants has not increased enough to treat such large quantities, so the untreated sewage is often discharged directly into rivers, leading to their pollution and increase in waterborne diseases. So because population is increasing so much, sewage also increases, and industrialization and urbanization has also taken place, and because of this, large quantities of sewage is being produced, but the wastewater or sewage treatment plants are the same number even now, that is they are not increasing with the increase in demand of sewage treatment plants. So therefore, most of the time a lot of people, they discharge this wastewater and sewage into rivers and other that is rivers and lakes and all, and this leads to the pollution of water, and it increases waterborne diseases. So because of this, the Ministry of Environment and Forest has initiated the Ganga Action Plan and the Yamuna Action Plan to save these major rivers in our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment plants so that only treated sewage may be discharged into the rivers. A visit to a sewage treatment plant situated in any place would be very interesting and educating experience. So because of such a large amount of sewage being discharged into sewage treatment plant, the Ministry of Environment and the Ministry of Forest has initiated the Ganga Action Plan and the Yamuna Action Plan. So under these plans, it is proposed to build a large number of sewage treatment plants so that only treated sewage can be discharged into rivers. Microbes in production of biogas. So we saw that in the secondary, that is the activated sludge which was formed in the secondary treatment. A part of it went as inoculum to the aeration tank, and the majority of it uh, went to the anaerobic sludge digesters, from which the anaerobic bacteria, it consumed all that flocks of aerobic bacteria and fungi, and it produced a lot of gases, namely methane, hydrogen disulfide, and carbon dioxide. Hydrogen sulfide, okay, H2S. Huh. And carbon dioxide. So this is basically as, and this produces biogas and microbes in production of biogas. Biogas is a mixture of gases containing predominantly methane, produced by the microbial activity, and which may be useful as may be used as fuel. So hydrogen sulfide, methane, and all, it is used as in the production of biogas, and this can be used as, it is a very good fuel, and especially with petroleum and all being fossil fuels and declining day by day, this biogas it can be used for cooking food and all, so it is really good. So now let's study about biogas. Biogas is a mixture of gases which contains predominantly methane, produced by the microbial activity, and it may be used as a fuel. You have learned that microbes produce different types of gaseous end products during growth and metabolism. The type of gas produced depends upon the microbes and the organic substrates they utilize. In the example cited in the relation to fermentation of dough, cheese making, production of beverages, the main gas was carbon dioxide, and what was involved, that is mainly, it was always yeast, that is Saccharomyces cerevisiae, which was used in the making of dough, that is bread making, cheese making, production of beverages such as wine, beer, rum, brandy, whiskey, and mainly carbon dioxide was produced. Other also, for cheese also, we saw Propionibacterium sharmani, which produced Swiss cheese, that is the holes were due to carbon dioxide in Swiss cheese. Then uh, so However, certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with carbon dioxide and hydrogen. Certain bacteria, they grow anaerobically, that is anaerobically without consuming oxygen, that is anaerobic respiration takes place on cellulosic material, and they produce large amount of methane along with carbon dioxide and hydrogen. These bacteria are collectively called as methanogens. One such common bacterium is methanobacterium. Methanobacterium, it produces methane. It is found in the ruminant, in we had learned before in Monera also, ruminant of in ruminant animals, in the gut of ruminant animals, and it helps in producing methane. That is, it digests cellulose to form methane anaerobically. Digests cellulose to produce methane anaerobically. These bacteria are commonly found in the anaerobic sludge during sewage treatment. So you know, in the anaerobic sludge digesters, when the activated sludge is sent, then the anaerobic bacteria, they digest or consume on this anaerobic sludge, and they form methane, hydrogen sulfide, and carbon dioxide. These bacteria are also present in the rumen of cattle, ruminant of gut of ruminant animals. These bacteria are also present in the ruminant or rumen, that is a part of stomach of cattle. A lot of cellulosic material present in the food of cattle are also present in the rumen. In rumen, these bacteria help in the breakdown of cellulose, and they play an important role in the nutrition of cattle. So in rumen, these bacteria, they help in the breakdown of cellulose, and they play an important role in the nutrition of cattle. Do you think we human beings are able to digest the cellulose present in our foods? No. Thus, the excreta or dung of cattle, commonly called as gobar, is rich in these bacteria. Dung can be used for the generation of biogas, commonly called as gobar gas. So we are not able to digest cellulose. We don't have that anaerobic bacteria, but the cattle, they do have these anaerobic bacteria called as methanogens, and therefore the excreta or dung of these cattle is commonly called as gobar, and this gobar is very rich in these bacteria called methanogens. Therefore, the dung of gobar, that is dung or gobar of these cattle, can be used for the generation of biogas, and this is commonly called as gobar gas. The biogas plant consists of concrete tank which is 10 to 15 feet deep, in which bio waste are collected and a slurry of dung is fed. Now let's see how a biogas plant works, that is, or how it looks. So biogas plant consists of a concrete tank, that is a tank which is made up of concrete cement, concrete tank, and it is 10 to 15 feet deep. So this concrete tank is 10 to 15 feet deep. It is 10 to 15 feet deep, and the bio waste are collected in a slurry of dung, in which bio waste are collected and a slurry of dung is fed. So this bio waste is collected in this 10 to 15 feet deep biogas plant, and a slurry of dung is fed. Slurry, that is basically in a liquid form. It is in a semi-solid form, or you can say not in a dry solid form. A floating cover is placed over the slurry. Over the slurry, a floating cover is placed which keeps on rising as the gas is produced. So floating cover is placed, so it keeps rising as the gas is produced. It is placed over the slurry in the tank due to microbial activity. Biogas plant has an outlet which is connected to a pipe supply biogas to nearby homes. So this is the pipe which is connected, and uh, this is the floating and this is the dung and water, that is slurry liquid form, it is gone over here in the digester. This is the biogas plant has an outlet. This is the which is connected to the pipe, and it supplies biogas to the nearby houses. The spent slurry is removed through another outlet which may be used as a fertilizer. So this is this is through which, this is the slurry, dung plus water. This is the slurry which goes over here where the biogas is produced. Here methane, carbon dioxide and hydrogen sulfide is formed, and this is the floating cover which keeps rising as this biogas is being formed, and this is the pipe which is connected which takes the biogas to nearby houses, and this is another outlet in which the slurry which has been used is removed to another outlet, and it may be used as a fertilizer. Cattle dung is available in large quantities in rural areas where cattle are used for a variety of purposes. So biogas plants are more often built in rural areas. Why? Because in rural areas there are many cows, goats and all. There are many cattle are there basically. So because over there agriculture and all is a major, so therefore in rural areas, since there is more cattle, there is more cattle dung and more gobar, so more gobar gas can be produced. So therefore majorly biogas plants are built in rural areas. So biogas thus produced is used for cooking and lighting, and the picture of a biogas plant is shown in figure 10.8. The technology of biogas production was developed in India mainly due to the efforts of Indian Agricultural Research Institute, AIRI, because of this, and Khadi and Village Industries Commission, KVIC, Khadi and Village Industries Commission, KVIC. So the technology of biogas was developed in India mainly due to the efforts of which particular, that is Indian Agricultural Research Institute, IARI, and Khadi and Village Industries Commission, KVIC. If your school is situated in a village or near a village, it would be very interesting to inquire how many biogas plants are nearby and visit them and learn more about it. Okay. Now microbes, biocontrol agents. Okay, so now let's see microbes as biocontrol agents. Biocontrol. What is biocontrol? Biocontrol refers to the use of biological methods. Sorry. Biocontrol refers to the use of biological methods for controlling plant diseases and pests. In modern society, these problems have been tackled increasingly by the use of chemicals, by the use of insecticides and pesticides. These chemicals are toxic and extremely harmful to human beings and animals alike, and they have been polluting our environment, that is soil, groundwater, fruits, vegetables and crop plants. Our soil is also polluted through our use of weedicides to remove weeds. Biological control of pests and diseases. In agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemicals. A key belief to the organic farmer is that biodiversity furthers health. The more variety a landscape has, the more sustainable it is. Okay. So what is biocontrol? Biocontrol refers to the use of biological methods for the control of plant diseases and pests. In modern society, how has these problems been tackled? They have been tackled by the use of insecticides and pesticides, but the use of insecticides and pesticides both are very harmful to plants as well as animals alike. To human beings and animals alike. In a way, even plants also. That's why disease-resistant plants are being made, and strategies for enhancement of food production we had learned, right? So now they have been polluting our environment, soil, groundwater, fruits, vegetables, and crop plants. Yeah, see, they have been polluting our crop plants, everything. Environment, fruits, vegetables, and all. Our soil is also polluted through the use of weedicides to remove weeds. So therefore, there is an important need for biological control of pests and diseases. Now, how do we do that? In agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemicals. So in agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemical. A key belief of the organic farmer is that biodiversity furthers health. Biodiversity furthers health. The more variety a landscape has, the more sustainable it is. The organic farmer therefore works to create a system where the insects are sometimes called pests are not eradicated, but instead are kept at manageable levels by a complex system of checks and balances within a living and vibrant ecosystem, contrary to the conventional farming practices which often use chemical methods to kill both the useful and harmful life forms indiscriminately. This is a holistic approach that seeks to develop an understanding of the webs of interaction between the myriad of organisms that constitute the field, fauna and flora. Organic farmer holds the view that eradication of the creatures that are often holistic approach. The organic farmer holds that eradication of the creatures that are often described as pests is not only possible but also undesirable, for without them the beneficial predatory and parasitic insects which depend upon them as food or host would not be able to survive. Thus the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. An important part of the biological farming approach is to become familiar with the various life forms that inhabit the field, predators as well as pests, and also their life cycle patterns of feeding and habits that they prefer. This will help appropriate means of biocontrol. In the feeding and the habits they prefer, this will help develop appropriate means of biocontrol. Okay. Now, so biocontrol refers to the use of biological methods for controlling pests, insects, pests, and controlling plant diseases and pests. In modern society, these problems have been tackled by increasing use of insecticides, pesticides, weedicide, and all. And these chemicals are very toxic, harmful to human beings. They pollute the crop plants as well as your soil, fruits, vegetables, etc. They are also very harmful to animals and all. So therefore, and soil gets polluted by the use of weedicides to remove weeds. Therefore, biological control of pests and diseases is necessary. In agriculture, there is a method of controlling pests. It relies on natural predation rather than chemicals and all. So, key belief of the organic farmer is that biodiversity furthers health. The more the biodiversity, the more that is the more variety a landscape has, more sustainable it is. So basically, if there is one disease-causing organism which let's say attacks your pea plant, so if you have pea plant plus your beans plus your tomato plus this plus that, so many things, so then and that too you have not grown it in a very uniform fashion. So the more variety, if otherwise if I will grow entire stretch same plant, then all of them will get affected at once. Whereas if I grow like this strip one plant, then this strip another plant, this strip another plant, then this strip again this one, the same one, so then the productivity will be also more, yield will be also more, plus there will be good so there are a lot of such as one is consuming more amount of nitrogen, one is giving more nutrient, one is a legume, so again nutrient वापस मिल रहा है उसको. So there are, you can do like that. So that is what organic farmer therefore works to create a system where insects, which are sometimes called pests, are not eradicated but instead kept at manageable levels by complex system of checks and balances within a living and vibrant ecosystem, contrary to the conventional farming practices which often use the chemical method to kill both the useful and harmful life forms indiscriminately. So if you are killing one microbe which is harmful and causing diseases, then along with it the useful microbe which was actually doing atmospheric fixation of nitrogen or something will also get killed. So this is a holistic approach that seeks to develop an understanding of the webs of interaction between myriad of organisms that constitute the field of flora and fauna. So basically you understand the entire interaction between the organisms and all. The organic farmer holds the view that the eradication of the creatures that are often described as pests is not only possible but also undesirable, for without them the beneficial predatory and parasitic insects which depend upon them as food or host would not be able to to survive. So if you eradicate one insect completely, then the bigger organism which used to feed on those insects will no longer have them. So basically, eradication of the creatures which are often described as pests is undesirable because without them, beneficial predatory and parasitic insects which depend upon them as food or host would not be able to survive. Thus, the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. An important part of biological farming approach is to become familiar with the various life forms. Otherwise, you will not be able to practice biocontrol, and that inhabit the field predators as well as pests, also their life cycle, pattern of feeding and habits that they prefer. This will help us to develop an appropriate means of biocontrol. The very familiar beetle with red and black markings, that is the ladybird, dragonflies, are useful to get rid of aphids and mosquitoes respectively. An example of microbial biocontrol agent that can be introduced in order to control butterfly caterpillars is the bacteria Bacillus thuringiensis, often written as BT. These are available in sachets as dried pores which are mixed with water and then sprayed to the vulnerable plants such as brassicas and fruit trees, where these are eaten by the insect larvae. In the gut of the larvae, the toxin is released and the larvae get killed. So the bacterial disease will kill the caterpillars but leave other insects unharmed. Because of the development of method of genetic engineering in the last decade or so, scientists have introduced Bacillus thuringiensis toxin genes into the plants, and such plants are resistant to attack by insect pests. BT cotton is one such example, which is cultivated in some states in our country. A biological control being developed for the use of treatment of plant disease is the fungus Trichoderma. Trichoderma species are free-living fungi that are very common in root ecosystem. They are effective biocontrol agents of several plant pathogens. Baculovirus. Baculovirus are pathogens that attack insects and other arthropods. The majority of baculoviruses are used as biocontrol agents are in the genus Nucleopolyhedrovirus. These viruses are excellent candidate for species specific narrow spectrum insecticidal application. They have been shown to have no negative impact on plants, mammals, birds, fish, or even non-target insects. This is especially desirable. Baculovirus, they usually show, I think, commensalism, right? They are neutral as well as they are positive or neutral, something like that. Or that is something different. We'll see in organism populations is yet to do. This is especially desirable when beneficial insects are being conserved to aid in an overall integrated pest management program IPM, or when an ecologically sensitive area is being treated. Okay. So the very familiar beetle with red and black markings, that is the ladybird and dragonflies, they are very useful to get rid of aphids and mosquitoes. Aphids, they show not Rhinotoki, not Philotoki, Amphitoki. There is parthenogenesis producing both males and females, Amphitoki. Example is aphids. Okay, and another example of microbial biocontrol agent that is to control butterfly caterpillars is Bacillus thuringiensis. Bacillus thuringiensis, often written as BT, is available in sachets as dried pores mixed with water and sprayed into vulnerable plants such as brassicas and fruit trees. When eaten by insect larvae, that is in the gut of the larvae, the toxin is released and the larvae then get killed. The bacterial disease will kill the caterpillars, but it will leave all the other insects unharmed. Because of the development of methods of genetic engineering in the last decade or so, we can introduce the Bacillus thuringiensis as a toxin gene into the plant, and such plants, for example BT cotton, are resistant to attacked by insect pests, and it is being cultivated in some states of our country. Another biological control which is being developed for the treatment of plant diseases is Trichoderma, which is a fungus. Trichoderma species are free-living fungi which are very common in root ecosystem. They are effective biocontrol in several agents of several plant pathogens. Now, baculovirus are pathogens which attack insects and other arthropods, and the majority of baculoviruses are used as a biocontrol agent. They come in the genus Nucleopolyhedrovirus. They are excellent candidates for species-specific narrow-spectrum insecticidal applications, and they have shown no negative impact on plants, mammals, and non-specific insects. birds, fish, etc. And they do not target them. They only target the ones that we term as undesirable and want to eradicate. So they are excellent insecticides. This is especially desirable when beneficial insects are being conserved to aid in overall integrated pest management program, or when an ecologically sensitive area is being treated. So why is this biocontrol agent especially desirable? Because when beneficial insects are being conserved to aid in overall integrated pest management program. So we have beneficial insects also, right? The ones which are pollinators, which help in pollination, or a lot of insects are there. So to conserve the beneficial insects in IPM, Integrated Pest Management Program, IPM, Integrated Pest Management Program, or when an ecologically sensitive area is being treated. When an ecologically sensitive area is being treated, that is the diversity of insects, and there are some kind of rare ones which we don't want to harm, because otherwise they are endangered already, they will go extinct or something. Then these baculoviruses are very useful, because they will only target specific insect and they won't harm the ones that are useful or beneficial to us human beings. Now, microbes as biofertilizers. With our present day lifestyles, environmental pollution is a major cause of concern. The use of chemical fertilizers to meet ever increasing demand of agricultural produce has contributed significantly to this pollution. So with our present day lifestyle, environmental pollution is major cause of our concern, and the use of chemical fertilizers to the ever increasing demand of agricultural produce has contributed significantly to this pollution. Of course, we have now realized that there are problems associated with the overuse of chemical fertilizers. There is a large pressure to shift to organic farming and use biofertilizers. So till now we have seen everything related to our human welfare only. First, we saw your lactic acid bacteria. Okay, wait. First, we saw your lactic acid bacteria, that is in our houses, how lactic acid bacteria helps us, how your baker's yeast, brewer's yeast, that is Saccharomyces cerevisiae, helps us, how toddy is being made by fermentation of sap from palms, and how other fermentation foods are being made from fermenting of bamboo shoots and soya bean, fish and all. Then we saw Swiss cheese made by Propionibacterium charmani. Then we saw Roquefort cheese, which is ripened by growing a specific fungi on it. Then we saw that industrial scale beverages and all are produced in large vessels called as fermenters, wherein your Saccharomyces cerevisiae brewer's yeast is used. That is in malted beverages and fruit juices and all, you ferment them and to produce ethanol. Then with or without distillation, that is without distillation, your wine and beer can be produced, and whiskey, brandy and rum is produced with this distillation of fermented broth. Then we saw another use of microbes, that is antibiotics, that is penicillin. It was discovered by accident by Alexander Fleming, and how Alexander Fleming, Ernest Chain and Howard Florey in 1945 got the Nobel Prize for it, and how antibiotics are help us to overcome many diseases such as plague, whooping cough or kali khasi, diphtheria or galgotu, and kush stroke or leprosy. Then we saw so after seeing microbes as your household products and as your antibiotics, now we saw them as organic acid, that is butyric acid by Clostridium botulicum. Then Acetobacter aceti, again another bacterium, that is as for production of acetic acid, and citric acid made by Aspergillus niger, which is a fungi, and lastly lactobacillus, which is a bacteria which helps in making lactic acid. After that is organic acids, we saw enzymes. What are the enzymes? That is lipases. Lipases are used in detergent to remove oily stain. Then we saw pectinases and proteases, which are used in clarification of bottled fruit sugars. Then after we saw streptokinase, which is used as a clot buster in people who are having myocardial infarction, which leads to heart attacks. So therefore their clots can be removed with the help of streptokinase, which comes from the bacteria Streptococcus. Then another bioactive molecule was cyclosporin A, which comes from Trichoderma polysporum. That is it is used as an immunosuppressive agent in organ transplant patients. Then statins is produced by another yeast which is called as Monascus purpureus. Monascus purpureus produces statins, which has been commercialized to lower blood cholesterol. It acts as a blood cholesterol lowering agent, because it competitively inhibits the enzyme which leads to the synthesis of cholesterol. Then after seeing as organic acid, as enzyme, and as bioactive molecules. Now microbes help us in sewage treatment plant, that is the municipal wastewater, also called as sewage, and sewage treatment plant STPs are present in all over our city, in our country, because we can't directly dispose the wastewater which contains human excreta and so many harmful pathogens and microbes into rivers and lakes directly. So therefore it has to be treated in sewage treatment plants first. So firstly comes the primary treatment, which involves filtration and sedimentation. So firstly by sequential filtration, that is all the floating debris is removed. Then by sedimentation, that is the grit is removed. What is grit? It consists of small pebbles and soil. This, what is this called? This is called as the primary sludge, and this settles down, and then the supernatant, that is above the primary sludge, whatever has not settled down, that is called as effluent. This effluent is then taken to for the secondary treatment of that is secondary treatment. Secondary treatment is also called as biological treatment. Why? Because it majorly involves the use of microbes and not any other mechanized something else. It mainly involves microbes. Then it is taken to an aeration tank, wherein mechanically oxygen is continuously being pumped, and useful aerobic microbes are grown in the form of flocs, which are joined by fungal filaments, and these flocs then consume the major part of the organic matter of the effluent, that is they start consuming oxygen. So therefore the BOD, that is biochemical oxygen demand, keeps getting lesser and lesser. The more the BOD of the wastewater, the more polluting it is. So these flocs, bacterial flocs, which are joined by fungal filaments, they basically lessen the biochemical oxygen demand of the wastewater, and therefore they make it less polluting. After this, they from the aeration tank, this is taken to a settling tank, in which these bacterial flocs is settled down, and these settled bacterial flocs, out of this, a portion of it is as inoculum or starter, it is sent to the previous aeration tank, wherein, like we saw in from curd to milk, the lactobacillus or lactic acid bacteria lab were used as inoculum or starter. Similarly over here, these bacterial flocs are used as inoculum or starter, so that in the aeration tank they again start growing and start consuming lessening the biochemical oxygen demand in the aeration tank. Now coming back to where we were in the settling tank, the major part or the remaining part of the bacterial floc which has settled down, it is called as activated sludge, and it is sent to anaerobic sludge digesters, where this aerobic bacteria and the fungi, that is these bacterial flocs, they are consumed by anaerobic bacteria, namely methanogens, which lead to the production of biogases, gases such as methane, hydrogen sulfide, and carbon dioxide. And methane, hydrogen sulfide, and carbon dioxide, these together are the constituents of biogas, and they they act as a biofuel, that is fuel in lighting and heating and all those things, that is cooking in people's houses. Now the effluent, that is in the secondary treatment plant, that is in the settling tank, the effluent, it is then released into the natural water bodies such as rivers and streams. Now, Ganga Action Plan and Yamuna Action Plan were done by the Ministry of Environment and Forest, because because with the increasing of pollution, urbanization, and industrialization, the sewage treatment plant number is still remaining constant or same. Therefore, there was a need for more, and therefore a lot of people discharge their sewage directly into water bodies and rivers, and therefore it led to huge pollution, which led to the Ministry of Environment and Forest to initiate a Ganga and Yamuna Action Plan, which leads to its purpose is to build a large number of sewage treatment plants, so that it can be treated, it can be discharged into the river. Time then so after sewage treatment plant, then biogas also, we saw that in the anaerobic sludge digesters, the anaerobic bacteria, which were called as methanogen, namely common bacterium name is methanobacterium. It is also found in the gut of ruminant animals, and it is basically helps in production of biogas, and that is, it feeds on cellulose in the gut of ruminant animals, and it forms biogas. Now Yeast was used in basically mainly yeast, and all other bacteria were used in the production of carbon dioxide. Similarly, over here, that is since in the gut of ruminant animal, methanogens feed on cellulose to form methane. Therefore, the Therefore, the dung of cattle, it is called as gobar, and this gobar contains methanobacteria, methanogens, and this helps in producing biogas, which is also called as gobar gas. Biogas plant that is 10 to 15 deep, made up of concrete. It has bio waste, that is slurry, dung plus water, that is liquid form or semi-solid form. That is a floating cover is there, which keeps rising as the biogas keeps forming. From one portion, that is the slurry, goes into the digester, and then the gas holder is there, and then the floating cover is there, and then a pipe is connected, which goes to the homes of people, used for cooking, heating, lighting, that is through which the biogas is transported. Then there is another used slurry comes out from to another outlet is there, and this used slurry is used as fertilizer, and it is very fertilizer manure, and it is very fertile rich. Now, usually these biogas plants are usually in rural areas. Why? Because cattle is more, so dung will be also more, gobar more, so gobar gas is also more. So therefore, mostly in rural areas these are made. And technology of biogas production was made by whom? Indian Agricultural Research Institute, IARI, and Khadi and Village Industries Commission, KVIC. Microbes as biocontrol agents. Biocontrol refers to the use of biological methods for the controlling of plant disease and pest. That is, if we use insecticide and pesticide, that is modern methods, chemical, then it is not good. It is extremely harmful to human beings, animals. It causes pollution in vegetable, fruit, and crop plants and all. And soil is also polluted by the use of weedicides to remove weeds. Therefore, biocontrol agent is very necessary, and biocontrol agent basically uh, works on understanding the interaction between the life forms, and it is a holistic approach, and it that is it aims to control the insect and pest by keeping a complex system of checks and balances instead of just spraying chemicals on it. And um, Another important form of biological is to become familiar with the life forms, their life cycles, patterns of feeding habit, etc. that they prefer. What are the main examples of biocontrol agent? That is, ladybird and dragonflies are help in getting rid of mosquitoes and aphids. Then Bacillus thuringiensis, it helps in getting rid of butterfly caterpillars. That is, it is available in sachets of dried pores mixed with water, sprayed into vulnerable plants such as brassicas and fruit trees. And when it is eaten by the caterpillar, then a toxin is released and the larvae gets killed in the gut of the larvae. Then somehow, that is, the Bacillus thuringiensis, that is, scientists have made BT cotton, that is, they have introduced these toxin genes into the plant itself, and therefore this plant BT cotton is resistant to these insect pests. Another biological control is trichoderma. That is, trichoderma is a free living species of fungus which lives in the root ecosystem, and they are effective biocontrol agent against several plant pathogens. Then baculovirus helps in attacking insect and other arthropods. It is used as a biocontrol agent in the genus Nucleopolyhedrovirus. Excellent candidate, species specific, narrow spectrum. Why is it very essential? Because in integrated pest management program, ecologically sensitive area is being treated. That is, all the beneficial pests and insects and all, they will all remain as it is, and this shows no effect on mammal, non-specific insect, pest and all. It only that is this baculovirus will only target species specific or very specific ones that we need to eradicate, only those microbes or pest and insects, sorry. Microbes as biofertilizers. Due to environmental pollution, that is organic farming is necessary. The use of biofertilizers is necessary. Biofertilizers are organisms which enrich the nutrient quality of the soil. Main source of biofertilizer are bacteria, fungi, cyanobacteria, that is in the roots of leguminous plants. Symbiotic association of rhizobium is seen. Rhizobium is a very important atmospheric nitrogen fixer. This bacteria fix atmospheric nitrogen into organic forms used by plant as nutrient. Other bacteria which can also fix atmospheric nitrogen while free living in the soil are spirillum and azotobacter, thus enriching the nitrogen content of the soil. So rhizobium lives in symbiotic association with the roots of leguminous plants. Rhizobium lives in symbiotic association, that is both have a positive effect, that is mutual relationship, that is the rhizobium will get something, whereas there is a plant will get nitrogen, rhizobium will get shelter. So rhizobium lives in symbiotic association with the roots of leguminous plants. Legumes or leguminous plants, that is all your pulses. Now free living, some other examples of free living bacteria which fix nitrogen are free living bacteria which fix nitrogen are azospirillum. Example is azospirillum, and azotobacter azospirillum and azotobacter. They also help in enriching the nitrogen content of the soil. Fungi can also form symbiotic association with plants. This is called as mycorrhiza. So fungi can also form symbiotic association with plants. This is called as mycorrhiza. Many members of the genus Glomus form mycorrhiza. Many members of the genus Glomus form mycorrhiza. Fungal symbiont in these association absorbs phosphorus from soil and passes it to the plant. The fungal symbiont, what does it do? It absorbs phosphorus from the soil and passes it to the plant. Plants having such associations, phycobiont, mycobiont, we had read in plant kingdom and biological classification. Plant having such association show other benefits also, such as resistance to root-borne pathogen, tolerance to salinity and drought, and an overall increase in the plant growth and development. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments. That is cyanobacteria, blue-green algae. They fix atmospheric nitrogen. Example is Anabena, nostoc, oscillatoria. In paddy fields, cyanobacteria serve as important biofertilizer. Blue-green algae also add organic matter to the soil and increase its fertility. Currently in our country, number of biofertilizers are available commercially in the market, and farmers use these regularly in their fields to replenish soil environments and reduce dependence on chemical fertilizers. So azospirillum, azotobacter, rhizobium, Anabena, nostoc and oscillatoria. All of these can fix atmospheric nitrogen. And fungi also form symbiotic relationships. Rhizobium forms symbiotic relationship with the roots of leguminous plant, and mycorrhiza, that is fungi, algae, they also form, that is fungi forms symbiotic association with the higher plants. Many members of the genus Glomus form mycorrhiza, and what does the fungal part do? That is the phycobiont, right? So the phycobiont, that is the fungal part, it absorbs phosphorus from the soil and passes it to the plant. And these plants also have other benefits after having such association, such as resistance to root-borne pathogen, tolerance to salinity and drought, and increase in plant growth and development. That's it for the chapter. Are there any important questions? Vitamin B12 increase करता है. Healthy also, it is good for digestion and all. Name some traditional foods. So many. Bread and all, it is involves the use of microbes like yeast. And then you can ferment rice to form beverages also with the help of yeast. You can control the disease caused by harmful in which way? Antibiotics. Name 2 species of fungus which are used for production of antibiotics. Penicillin, then ampicillin. What is sewage? That is municipal wastewater. Municipal wastewater. In which way can sewage be harmful to us? It has pathogens. Human excreta. Primary and secondary. Primary basically involves that is sedimentation and filtration, whereas secondary sewage treatment is also called as biological sewage treatment because it involves the use of microbes. Yes, microbes can be used as source of energy because biofuel, that is biogas. Methanobacterium, it can be that is, it is an anaerobic bacteria which on that is converts your slurry, that is dung and water, that is slurry, into gobar gas by feeding on by feeding on aerobic bacteria and fungi, flocks of bacteria. Microbes can be used to decrease the use of chemical fertilizers and pesticides. As biocontrol agents, they can act, such as Bacillus thuringiensis, then your Trichoderma, and अच्छा microbes, हाँ microbes में यही आएंगे. Then your Rhizobium, Anabaena, Nostoc, Oscillatoria, Acetobacter, Azospirillum. Then most polluted. VOD values are 28, 400. 400 is most polluted. To each assuming the, can you assign the correct label to each assuming the river water is relatively clean? River water and untreated sewage. Acha. River is relatively clean, so this will be river. Untreated sewage will be this, and secondary effluent will be this. This is secondary effluent. This is river water. Because it has said that the river water is clean, and this is untreated sewage. Untreated sewage. So over here the biochemical oxygen demand is relatively lesser because it is secondary sewage, because aerobic bacteria and aeration tank have been feeding on it, whereas in river water it is completely less because that process is over, whereas over here it is untreated sewage, so BOD is very high. Find out the name of the microbes from which cyclosporin A, Trichoderma polysporum. Trichoderma polysporum. And statins. Statins is Monascus purpurus. Monascus purpurus. Which is this is a fungi. Blood cholesterol lowering agent. I think, yeah, yeast, a form of yeast. This is also fungi. Find out the role of microbes. Single cell protein and enhancement in food production. We had learned that spirulina and all, that is spirulina and with Hylotropas can be grown in on that is molasses and wastewater and all, and they produce a large amount of protein, and they can be a very good protein alternative source of protein because in order to eat 1 kg of meat, a lot of grain has to be 5 to 10 kg of grain has to be gone to feed the cattle and the bull and the buffalo for, let's say, beef or meat or everything, that is goats also for mutton, and also it is with the increasing demand for protein and meat and all, it is not able to keep up, so therefore single cell protein was introduced. In soil, that is enrichment of soil, that is these all microbes, they help in enriching the soil, that is replenishing nutrients, nitrogen and all. For example, your rhizobium, arabina, nostoc and all, oscillatoria and all, free living ones, not rhizobium. Rhizobium free living ones was arabina, nostoc, oscillatoria, azospirillum and azotobacter. Arrange the following in the decreasing order, most important first for the welfare, give reasons for your answer. Penicillin is very important. Then biogas is also very important. Penicillin is very important because it's antibiotic, that is firstly your gums health. Then biogas, because it acts as a fossil fuel, it is very important with the declining of fossil fuel. Then curd and citric acid. How do fertilizers enrich the fertility of the soil? That is, they replenish the nutrients. Biofertilizers, for example, if you take rhizobium, then it is in symbiotic relationship with the it fixes that is nostoc, anabena, oscillatoria, which are cyanobacteria, blue-green algae, and azospirillum, azotobacter, and rhizobium. All of these, they fix atmospheric nitrogen, and they replenish the soil. So with that, we are done. Thank you.